You think about picking up your coffee cup, or typing a text message, or even just taking a breath, and your body instantly obeys.
Right. It just happens.
Yeah, and I feel like we tend to get completely lost in the abstract when we talk about the brain. We discuss consciousness, memory, and emotion as if they exist in a vacuum.
Oh, absolutely. We treat the brain like a computer floating in space.
Exactly. But the reality is that the central nervous system is an engineering marvel designed with a very specific physical output in mind. Every single one of those actions you take requires a physical translation of thought into mechanical force.
The abstract really has to become concrete. I mean, the electrical impulse generated in the motor cortex must travel down the spinal cord, exit the central nervous system, and ultimately trigger a mechanical contraction.
It's the only way we actually do anything.
Literally. Movement is the sole mechanism by which we interact with our environment. The challenge of deciding when and how to move is, to a massive degree, the primary driving force behind the entire evolution of the nervous system.
That actually brings to mind a quote from the pioneering neurophysiologist Charles Sherrington. Writing all the way back in 1924, he noted that to move things is all that mankind can do. For such the sole executant is muscle, whether in whispering a syllable or in felling a forest.
That is a phenomenal quote, and it perfectly frames what we are looking at today.
It really does. So welcome to this special deep dive tailored specifically for the Enkephalikos channel. We are embarking on part 1 of a 3-part series, systematically deconstructing Chapter 57 of Principles of Neuroscience.
Yes, and we're looking intimately at the diseases of the peripheral nerve and motor unit.
Right. Our mission today is to tear down the biological machinery of movement to understand how it operates. We want to see how a neurologist plays clinical detective to locate a failure in the system and thoroughly analyze the devastating diseases that target our motor neurons and peripheral nerves.
But to understand the pathology, we first have to understand the baseline architecture. I mean, Sherrington introduced a fundamental concept just a year after he wrote that quote.
In 1925, right?
Yeah, exactly. In 1925, he coined the term motor unit. A motor unit is defined as a single motor neuron and the specific group of muscle fibers it innervates.
So you can think of the motor neuron sitting inside the spinal cord or brainstem as the commander.
Yes, the commander issuing the orders.
And its axon is the communication wire extending outward, and the muscle fibers are the troops that execute the specific command to contract.
That's a perfect way to visualize it. And the text makes it clear that the architecture of this unit varies wildly depending on the job it needs to do.
It's a classic case of form-following function, isn't it? The nervous system is constantly balancing precision against raw power.
Exactly. If you look at the muscles controlling your eye movements, those require exquisite microscopic control to track a moving object. So in the extraocular muscles, a single motor unit might contain fewer than 100 muscle fibers.
So the commander is directing a highly specialized, very small squad.
Right. But if you look at a muscle in the human leg, like the gastrocnemius, the requirements change completely. The leg doesn't require microscopic precision.
No, it requires bulk power. It has to propel your entire body mass against the force of gravity.
Right, so a single motor neuron innervating the leg muscles might control up to 1,000 individual muscle fibers. A massive battalion responding to a single electrical impulse.
Oh, wow, 1,000 to 1. That's a huge difference.
It is. But what is truly remarkable about this arrangement is the rigid biological rule governing the makeup of these units. We classify human skeletal muscle fibers broadly into different histochemical types based on their biochemistry.
Like the aerobic versus anaerobic fibers, right?
Exactly. Type I fibers are aerobic, packed with mitochondria and oxidative enzymes, making them highly resistant to fatigue. They're perfect for maintaining posture.
And then type 2 fibers are anaerobic, rich in glycolytic enzymes. They allow for rapid, powerful bursts of force, but they fatigue really quickly.
Right, and the fascinating rule here is the principle of uniformity. Because a single motor neuron dictates the biochemical properties of the muscle fibers it connects to, every single muscle fiber within one motor unit will be identical.
Wait, really? So the commander never directs a mixed unit?
Never. The neuron literally dictates the gene expression of the muscle fibers it innervates, forcing them all to become type 1, or forcing them all to become type 2.
That is incredible. And the nervous system also manages these different units with such elegance.
Yeah.
When you decide to initiate a movement, the motor units are not activated randomly, are they?
No, not at all. They are recruited in a fixed, highly specific sequence dictated by the size of the motor neuron cell body. The nervous system always recruits the smallest motor units first.
And those small neurons generally innervate the slow-twitch, fatigue-resistant type I bifibers, right?
Exactly.
I always visualize this like a symphony choir. If the conductor wants a very quiet, delicate melody, they don't have the entire massive chorus sing at a whisper. They start with a single, small soloist.
That's a great analogy.
And as the music needs to get louder, as more muscular force is required to lift a heavier object, the central nervous system brings in progressively larger sections of the choir. It recruits the larger motor neurons that innervate the powerful type 2 fibers.
Right, adding them to the contraction until the muscle is booming for the crescendo. The underlying physics of that recruitment order is rooted in electrical resistance.
Because smaller motor neurons have a higher input resistance.
Yes, meaning a smaller synaptic current is required to depolarize them to threshold. They naturally fire first.
Okay, so the biological machinery of movement has 4 highly distinct anatomical components. The motor neuron cell body in the spinal cord, the peripheral axon acting as the communication wire with its myelin insulation,
The neuromuscular junction where the chemical signal crosses the gap.
and the muscle fibers themselves.
Right.
And because the motor unit is built from those 4 specific functional components, it is predictably vulnerable in 4 distinct domains. So if a patient walks into a neurology clinic complaining of weakness, the clinician's job is to figure out which of those 4 domains is broken.
Precisely. The diagnostic process begins with a crucial first approximation, determining whether the weakness is neurogenic, meaning a problem with the nerve, or myopathic, meaning a primary disease of the muscle itself.
And the initial clue there is heavily topographical, isn't it?
It is. As a general rule, neurogenic disorders manifest primarily as weakness in the distal limbs.
Like the hands and the feet.
Exactly. The pathological changes begin at the farthest reaches of the peripheral nervous system. Primary muscle diseases, or myopathies, on the other hand, usually present with weakness in the proximal limbs.
So the shoulders, the hips, the thighs. A patient with a myopathy might describe immense difficulty climbing a flight of stairs or getting up from a low chair.
Right, or even just lifting their arms to wash their hair. To really grasp the neurogenic side, the text presents the starkest possible model, a completely severed peripheral nerve.
Let's talk about that. When a nerve is cut, the electrical commands are instantaneously halted. The muscle becomes totally paralyzed.
Yes, but the secondary effects are just as profound. The muscle rapidly begins to undergo severe atrophy.
And historically, the term atrophy literally meant a lack of nourishment.
Exactly. We now understand that the muscle physically wastes away because it requires the constant trophic chemical signaling and electrical activity from the nerve just to maintain its structural mass.
So it's not just sitting there idle, it's actually actively starving without the nerve.
Yes. The severed nerve model also highlights that peripheral nerves are bundled cables carrying two-way traffic. They contain motor axons sending descending commands out to the muscle, but they also contain sensory axons bringing tactile, pain, and thermal information back to the central nervous system.
So a severed nerve results in an immediate loss of sensation in the territory innervated by that specific nerve.
Right. Furthermore, the deep tendon reflexes are lost instantly because the reflex arc, that closed loop between the sensory spindle fiber input and the motor neuron output, has been physically severed.
Wow. Contrast that extreme nerve injury with the red flags of a myopathy. Alongside that proximal weakness, we look for signs like myotonia.
Right, myotonia. That is a state where the muscle fibers are hyperexcitable and literally cannot relax after a voluntary contraction.
So a patient might shake your hand and find their fingers locked in a grip, unable to let go.
Exactly. Patients also present with frequent cramps, myalgia, which is muscle pain, and an alarming clinical sign called myoglobinuria.
I wanted to bring that up. If muscle fibers are actively degenerating and bursting open due to a severe myopathy, they spill myoglobin directly into the bloodstream, right?
Yes, and myoglobin is the heme-containing protein that binds oxygen and gives muscle its red color.
And then the kidneys eventually filter that excess myoglobin, turning the patient's urine a dark, muddy red or brown.
Which is obviously a terrifying thing for a patient to see. So clinical signs give us the topography. But neurologists deploy a highly sophisticated electrodiagnostic toolkit to examine the internal electrical characteristics of the motor unit in real time.
Needle electromyography. EMG. This is such a fascinating procedure.
It's a cornerstone of the diagnostic process. The clinician inserts a fine recording electrode directly into the belly of the skeletal muscle.
So the needle is literally picking up the extracellular electrical potentials generated by the muscle fibers themselves.
Exactly. We evaluate the muscle in 3 distinct states: upon needle insertion while the muscle is completely at rest, and during varying degrees of voluntary contraction.
Let's establish the normal baseline first. In a healthy muscle, when the patient is completely relaxed, the EMG monitor is absolutely quiet, right?
Yes, completely quiet. There is no electrical activity outside of the microscopic noise right at the neuromuscular junction.
And then, when the patient is asked to make a very weak voluntary contraction, the needle picks up the firing of a single motor unit. The monitor displays crisp, discrete electrical spikes.
Right, and as the patient pushes harder, recruiting more and more units according to that choir principle we discussed, the potentials start to overlap.
Until at maximal contraction, so many units are firing simultaneously that the screen fills with a chaotic, dense thicket of electrical activity. The text calls it a profuse interference pattern.
You can no longer distinguish individual spikes, and the diagnostic power of the EMG becomes apparent when that baseline changes.
Like in a neurogenic disease.
Right, in a neurogenic disease where motor axons are dying, the resting muscle is no longer quiet. The EMG detects spontaneous low-amplitude electrical discharges known as fibrillation potentials.
And the biophysical mechanism here is wild. When a muscle fiber loses its nerve supply, the acetylcholine receptors, which are normally densely clustered only at the neuromuscular junction, begin to spread out.
Yes, they populate the entire surface of the muscle fiber. The sarcolemma, the muscle cell membrane, becomes globally hyperexcitable. The orphan muscle fiber begins to spontaneously depolarize and twitch on its own.
But these fibrillations are completely invisible to the naked eye because they are just single cells firing.
Exactly. But when the patient tries to voluntarily contract this denervated muscle, the EMG reveals the true extent of the damage. Because a large number of motor axons have died, the total number of motor units available for voluntary control is severely reduced.
So during a maximal contraction effort, instead of generating that dense, noisy interference pattern, the EMG only shows a sparse pattern of discrete, isolated motor unit potential.
Right. And here's where it gets really interesting. The potentials of the surviving units in a chronic neurogenic state often display a massive increase in amplitude and duration.
I was going to ask about that. If the nerve is dying, why do the electrical potentials of the surviving units sometimes get larger and longer?
It reflects the nervous system's incredible capacity for structural adaptation through a process called axonal sprouting.
Okay, so when a motor neuron dies, all the muscle fibers it innervated are suddenly abandoned.
Yes, and a neighboring healthy motor neuron senses the distress signals from these orphan fibers. The healthy neuron sprouts new axonal branches, reaching out to adopt and reinnervate the denervated muscle cells.
Oh, I see. The surviving commander's battalion just got exponentially larger. It has created a giant motor unit.
Exactly. So when that single surviving neuron fires, it depolarizes a far greater number of muscle fibers than it normally would, producing a massive electrical spike on the EMG.
And this process also explains a physical sign that clinicians can actually see with the naked eye, right? Fasciculations.
Yes. Unlike fibrillations, which are microscopic single-fiber twitches, a fasciculation is the spontaneous, involuntary contraction of an entire motor unit all at once.
You can actually see it as a visible ripple or flicker under the skin.
Right. The sick, dying motor neuron is electrically unstable, and its nerve terminal misfires, triggering the whole newly expanded squad simultaneously.
Okay, so that's the neurogenic picture. But the EMG picture for a myopathic state is entirely different, isn't it?
Entirely different. In a primary muscle disease, the nerve is perfectly healthy, but the muscle fibers themselves are degenerating. Because the nerve connection is intact, the resting muscle is usually quiet. You rarely see fibrillations.
So when the patient contracts the muscle, the normal number of motor units fire, the commander is issuing orders, and all the individual squads are receiving the signal. The interference pattern is fully present.
It is present, yes. But the squads have taken massive casualties. Individual muscle fibers within each unit have died or shrunken.
Oh, so consequently the electrical potentials generated by these depleted units are much smaller in amplitude.
Exactly. They're also of shorter duration and highly polyphasic, meaning the electrical wave has a jagged, complex shape with multiple alternating positive and negative peaks.
So the nerve is shouting its command, but the depleted muscle simply lacks the physical mass to generate a forceful or electrically robust response.
You nailed it. Electromyography evaluates the muscle's response. But we also must evaluate the integrity of the communication wire itself using nerve conduction studies.
Right, because this allows us to mathematically distinguish between diseases that strip the insulation off the nerve and diseases that destroy the central wire.
Exactly. We stimulate a motor nerve with a mild electrical shock, proximally for example, at the elbow, and record the time it takes for the thumb muscle to twitch. We then stimulate the nerve distally at the wrist and record that time.
And by subtracting the distal latency from the proximal latency and dividing by the physical distance between the two stimulation sites, we calculate the exact conduction velocity of the nerve impulse.
And that speed tells us precisely what structure is failing. The rapid propagation of an electrical signal down an axon relies on the myelin sheath. Myelin acts as a biological insulator, allowing the electrical current to jump rapidly from one uninsulated node of Ranvier to the next in a process called saltatory conduction.
So if a disease attacks the myelin, the current leaks out, and the conduction velocity drops dramatically. We call these demyelinating neuropathies.
Right, but if a disease targets the axon itself, the structural wire inside the insulation, the remaining intact axons still conduct at normal speeds.
However, because there are fewer wires carrying the signal, the overall amplitude of the electrical response drops. So slow speed indicates myelin damage, while low amplitude with normal speed indicates axonal damage.
That's the core of it. Now, the clinical detective work also relies on serological testing, specifically measuring the enzyme creatine kinase, or CPK.
And CPK plays a critical role in energy metabolism within the muscle cell sarcoplasm, right? Facilitating the rapid regeneration of ATP.
Yes. In a healthy individual, very little CPK leaks into the blood. But in a myopathy, the structural integrity of the muscle fiber membrane breaks down. The necrotic muscle cells rupture and dump their intracellular contents, including high concentrations of CPK, into the bloodstream.
So a significantly elevated serum CPK level is a major biochemical indicator of active muscle destruction.
Exactly. The final definitive piece of the diagnostic puzzle is the muscle biopsy, physically examining a cross-section of the tissue under a microscope.
And this brings us back to the rule of uniformity we established earlier. A healthy muscle contains a mosaic of overlapping type I and type II motor units.
Right. When stained for their specific oxidative or glycolytic enzymes, a healthy biopsy looks like a random, evenly distributed checkerboard of light and dark fibers.
But that pristine checkerboard is completely obliterated in a chronic neurogenic disease, isn't it?
It is. The axonal sprouting we discussed physically rewires the tissue. When a surviving neuron branches out to adopt denervated muscle fibers, it forces them to change their biochemical expression to match its own histochemical type.
Right, so a type I motor neuron will force adopted type II fibers to become type I. Over time, this creates massive, solid clusters of identical fiber types.
Yes. Under the microscope, you no longer see a checkerboard. You see huge, solid patches of type I fibers sitting adjacent to huge, solid patches of type II fibers. The text refers to this phenomenon as fiber type grouping.
It's basically incontrovertible histological evidence that a nerve has been slowly dying and a surviving nerve has been aggressively re-innervating the orphaned tissue.
Precisely. And the tragic conclusion of this process occurs if the disease relentlessly progresses and that heroic surviving neuron eventually succumbs and dies.
Oh, man. When it fails, that entire massive grouped cluster of identical muscle fibers loses its nerve supply all at once.
Exactly. The entire group undergoes denervation atrophy simultaneously. This histological pattern, known as group atrophy, is the absolute hallmark of advanced end-stage chronic neurogenic disease.
Whereas a myopathy, by contrast, shows random, scattered degeneration and regeneration of individual fibers, right? Often accompanied by inflammatory cell infiltration or the replacement of dead muscle tissue with fat and connective tissue.
Right. Now, before we delve into specific diseases, we have to establish one more critical neuroanatomical distinction: the difference between upper motor neurons and lower motor neurons.
Yes, this is crucial. Everything we have discussed so far, the motor neuron cell body sitting in the anterior horn of the spinal cord, sending its axon out to the skeletal muscle, is the lower motor neuron.
It is the final common pathway for all movement. But the lower motor neuron does not act independently. The lower motor neurons are entirely subservient to the upper motor neurons.
And the cell bodies of the upper motor neurons reside in the motor cortex of the brain. Their long axons travel deep into the brain, down through the brainstem, and descend through the spinal cord via the corticospinal or pyramidal tracts.
Right, they synapse onto the lower motor neurons, issuing the high-level commands for voluntary movement and constantly providing a modulating inhibitory influence over the lower circuits.
And clinicians can actually differentiate a failure in the upper pathway from a failure in the lower pathway simply by observing the patient's reflexes and muscle tone.
It's one of the most elegant parts of the neurological exam. Damage to the lower motor neuron physically disconnects the muscle from the nervous system. The muscle becomes flaccid. It loses its tone. It undergoes profound atrophy. You see the visible fasciculations, and the deep tendon reflexes are completely abolished because the reflex arc is broken.
But damage to the upper motor neuron produces an entirely different clinical picture.
Entirely different. The lower motor neuron and the reflex arc remain structurally intact, but they have lost the descending inhibitory control from the brain. The lower motor neurons become wildly hyperactive.
Which manifests clinically as spasticity, right? That state of tight, rigid muscle tone that is dependent on the velocity of movement.
Exactly. The deep tendon reflexes become exaggerated and hyperresponsive. The loss of upper motor neuron control also unmasks primitive, abnormal reflexes.
The most famous of these is the Babinski sign.
Yes. Normally, stroking the sole of the foot causes the toes to curl downward in flexion. In a patient with upper motor neuron damage, the big toe abnormally extends upward and the other toes fan out.
Okay, so equipped with this understanding of the clinical topography, the EMG, conduction velocities, and upper versus lower neuron signs, we can analyze the first major category of pathology, diseases that target the motor neurons themselves.
And the defining characteristic of these diseases is an incredible phenomenon called selective vulnerability. These pathological processes exclusively target and destroy motor neurons while completely ignoring adjacent sensory neurons, corner neurons, and the neurons of the autonomic nervous system.
The most notorious and devastating of these conditions is amyotrophic lateral sclerosis, widely known as ALS or Lou Gehrig's disease. The anatomical nomenclature of the disease maps perfectly to its underlying pathology.
It really does. Amyotrophy literally translates to a lack of muscle nourishment. It describes the profound, visible neurogenic muscle wasting, weakness, and fasciculations that occur as the lower motor neurons in the spinal cord and brainstem systematically die off.
And the second half of the name, lateral sclerosis, refers to the upper motor neuron component. The corticospinal tracts carry the axons of the upper motor neurons down through the lateral columns of the spinal cord.
Right. As these upper motor neurons degenerate, the support cells of the central nervous system, the astrocytes, proliferate in the lateral columns to clean up the damage, replacing the neural tissue with dense, fibrous glial scarring.
Sclerosis means hardness. Early pathologists performing autopsies could physically feel the abnormal hardness of the scarred lateral columns when sectioning the spinal cord.
Exactly. ALS is a combined neurodegenerative strike, destroying both the upper and lower motor pathways simultaneously. The clinical presentation is insidious and relentless. ALS typically strikes individuals in their 40s or 50s.
And it often begins focal and asymmetric, usually presenting as painless weakness in a single distal limb.
Yes. The patient might notice a sudden clumsiness in fine motor tasks, difficulty buttoning a shirt, turning a key, playing an instrument, or typing.
And the clinical examination at this early stage often reveals a cruel paradox. Wasting and fasciculations of the intrinsic hand muscles indicating lower motor neuron death, occurring simultaneously with hyperactive tendon reflexes in that same arm, indicating upper motor neuron damage.
It's devastating. From that focal starting point in the hand or foot, the disease spreads inexorably. Over the course of 3 to 4 years, the progressive weakness marches up the limb, crosses over to the opposite side, and ascends the spinal cord.
It eventually compromises the bulbar motor neurons in the brainstem, destroying the patient's ability to chew, swallow, and speak. Ultimately, the degeneration reaches the motor neurons controlling the diaphragm and intercostal muscles, leading to respiratory failure.
Yet, within this systematic devastation lies one of the most profound mysteries in neuroscience, bringing us back to selective vulnerability. Even as ALS destroys the motor control of the limbs and the respiratory system, it completely spares specific populations of motor neurons.
Like the motor neurons controlling the extraocular muscles of the eye, they remain perfectly intact. Patients in the advanced, locked-in stages of ALS can still track objects and communicate through eye movements.
Exactly. Furthermore, the disease spares the motor neurons in the sacral spinal cord, known as Onuf's nucleus, which maintain voluntary control over the bowel and bladder sphincters.
Wait, let me challenge that. If the most common forms of ALS are driven by genetic mutations, meaning the defective gene is present in the DNA of every single motor neuron in the body, why would the ocular motor neurons in Onuf's nucleus be immune to a mutation they actively carry?
That is the core question driving modern ALS research. It strongly implies that different populations of motor neurons possess fundamentally unique biochemical or metabolic signatures.
Oh, I see. So researchers hypothesized that the spared motor neurons might have specialized calcium buffering proteins that protect them from excitotoxicity.
Right. Or perhaps they lack certain membrane receptors that render the vulnerable spinal motor neurons susceptible to damage. They might handle oxidative stress more efficiently, or process toxic protein aggregates differently.
So the mutation is ubiquitous, but the cellular environment dictating whether that mutation is lethal is highly specific. Unlocking the precise molecular mechanisms behind this resistance could provide the key to neuroprotective therapies for the vulnerable neurons.
Exactly. And the genetic discoveries surrounding ALS have completely transformed our understanding of the cellular pathology. About 90% of ALS cases are sporadic, occurring with no clear family history. However, the remaining 10% are familial, generally inherited in an autosomal dominant pattern.
And by studying the genetic errors in that 10%, researchers have illuminated the molecular mechanisms driving the disease process across all forms of ALS.
Yes. The most common genetic culprit identified is a mutation in the C9orf72 gene. The nature of this mutation is structurally fascinating. It is an intronic hexanucleotide repeat expansion.
So inside a non-coding region of the gene, an intron, there is a sequence of 6 nucleotides: guanine, 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 cytosine, cytosine, or GGGCC.
Right. In a healthy genome, this 6-letter sequence might repeat 20 or 30 times. In a patient with this mutation, the DNA replication machinery stutters and expands that sequence massively, resulting in hundreds or even thousands of repeats.
And this massive genetic expansion triggers a catastrophic sequence of events within the cell, often described as a toxic trinity. The first element is a simple loss of function.
Yes, the sheer size of the expanded intron disrupts the normal transcription and processing of the C9orf72 gene, leading to depleted levels of the normal protein, which plays a critical role in vesicular trafficking and autophagy, the cell's waste disposal system.
And the second element of the trinity is RNA toxicity.
Exactly. When the cellular machinery transcribes this massive repetitive DNA sequence, it produces abnormally long, structurally complex RNA molecules. These mutant RNA transcripts accumulate in the nucleus and act like biochemical sponges.
So they sequester and permanently trap essential RNA binding proteins, preventing them from performing their normal regulatory functions, which severely disrupts the neuron's overall RNA metabolism.
Right. And the third piece of the toxic trinity involves a bizarre error in protein translation. Even though the expansion is located in a non-coding intron, the cellular machinery occasionally misreads the RNA through a process called repeat-associated non-AUG translation.
It blindly translates the repeating RNA into dipeptide repeat proteins, long, toxic chains of alternating amino acids such as polyglycine proline.
And these repetitive proteins are intrinsically neurotoxic and form dense aggregates within the cell. The theme of protein misfolding and aggregation unifies almost all the genetic forms of ALS.
The text details mutations in the SOD1 gene, which encodes copper-zinc superoxide dismutase, a vital antioxidant enzyme. Mutations destabilize the SOD1 protein, causing it to misfold and clump together, exposing toxic domains that generate severe oxidative stress.
We also see mutations in TDP43 and FUS. These are critical RNA-binding proteins that normally shuttle between the nucleus and the cytoplasm to regulate gene expression.
The mechanics of how TDP43 and FUS fail introduce the concept of stress granules. Under normal conditions, when a cell is exposed to environmental stress like heat or toxins, it halts normal protein production and rapidly packages its vital RNA and proteins into dense, membraneless organelles in the cytoplasm called stress granules.
They basically act as temporary biological safe houses to protect the cellular machinery until the stress passes, at which point the granules dissolve and normal function resumes.
But in ALS, the mutant TDP43 or FUS proteins become permanently entangled within these stress granules, don't they?
They do. The temporary safe houses undergo a pathological phase transition. They solidify into permanent, insoluble aggregates. These aggregates physically disrupt the intracellular architecture, trap functional proteins, and severely impair the motor neuron's ability to maintain its massive axonal volume, ultimately leading to cell death.
But the clinical progression of ALS, the way the weakness starts in one hand and predictably marches up the arm and across to the other limb, suggests something more sinister than individual cells randomly succumbing to genetic stress. If the protein aggregates are forming inside individual motor neurons, how does the pathology spread geographically through the nervous system?
Extensive research over the past decade points to a prion-like mechanism of cell-to-cell transmission. Prions, infamous for causing diseases like Creutzfeldt-Jakob or mad cow disease, are corrupted proteins that can induce healthy proteins to misfold.
So in ALS, evidence suggests that the toxic aggregates of SOD1 or TDP43 can be actively released or leak out of a dying motor neuron into the extracellular space, potentially packaged within small vesicles called exosomes.
Exactly.
And these corrupted proteins are then absorbed by the neighboring healthy motor neuron.
Precisely. Once inside the cytoplasm of the adjacent neuron, the misfolded aggregate acts as a corrupted structural template. It physically interacts with the cell's native healthy proteins and forces them to adopt the misfolded conformation.
So the newly corrupted proteins aggregate, the cell eventually dies, and the cycle repeats. This templated misfolding perfectly explains the contiguous anatomical spread of the disease through the connected networks of the spinal cord and motor cortex.
It does. But the textbook also highlights that motor neurons do not exist in isolation. The disease process heavily involves non-cell autonomous factors, emphasizing the role of the glial cells, the microglia and astrocytes that surround and support the neurons. Microglia are the resident immune cells of the central nervous system.
Initially, when motor neurons begin to suffer from aggregate toxicity, the microglia and astrocytes mount a neuroprotective response. They activate to clear out debris and release neurotrophic factors to support the struggling neurons.
However, as the disease progresses and the aggregates accumulate, this support system catastrophically fails. The microglia become chronically pathologically activated. They shift from a protective state into a neurotoxic state, pouring out inflammatory cytokines and reactive oxygen species that drastically accelerate the death of the motor neurons.
And the genetics link this inflammatory failure to other neurodegenerative diseases. The text points to variants in the TREM2 gene. TREM2 is a receptor expressed on the surface of microglia that regulates their activation and their ability to phagocytize or eat cellular debris.
Right, and certain genetic variants impair the function of TREM2, crippling the microglial response to injury. Crucially, these same TREM2 variants that increase the risk of developing ALS have also been shown to significantly increase the risk of developing Alzheimer's disease.
The survival of the central nervous system heavily depends on the precise, coordinated action of its immune support cells.
Absolutely. Now, while classic ALS is the most prominent motor neuron disease, the clinical spectrum includes variants with restricted pathology. Progressive bulbar palsy, for example, is characterized by the selective degeneration of the motor neurons located in the brainstem, which innervate to cranial nerves.
And the term BOLD is an archaic anatomical reference to the medulla and pons. Patients primarily present with severe dysarthria, an inability to articulate speech and dysphagia, and inability to swallow, while limb strength remains relatively preserved early in the disease.
Exactly. The final motor neuron disease the text extensively covers is progressive spinal muscular atrophy, or SMA. SMA is profoundly different from ALS. It restricts its damage entirely to the lower motor neurons in the spinal cord. The upper motor neurons in the brain remain completely unaffected.
Consequently, there is no spasticity. Furthermore, it is a developmental disorder that predominantly strikes infants and young children, presenting as profound symmetrical muscle weakness, hypotonia or floppiness, and absence of deep tendon reflexes and severe respiratory compromise.
The genetic basis of SMA provides one of the most fascinating examples of human evolutionary quirks in molecular medicine. SMA is an autosomal recessive disorder caused by mutations or deletions in the survival motor neuron 1 gene, or SMN1, located on chromosome 5.
And the SMN protein is ubiquitously expressed in all cells and is absolutely essential for the assembly of the spliceosome, the cellular machinery responsible for editing raw RNA into the final messenger RNA used to build proteins.
But if the SMN protein is essential for every cell in the body, why does losing it exclusively kill the lower motor neurons in the spinal cord? It's another profound example of selective vulnerability.
The extreme metabolic demands of maintaining a massive peripheral axon make the lower motor neuron exquisitely sensitive to any disruption in RNA splicing. But the human genome has a built-in failsafe. We possess a nearly identical copy of the SMN gene known as SMN2.
Yes.
I always visualize this setup like a hospital's electrical grid. The SMN1 gene is the primary nuclear power plant, generating 100% of the energy needed. If an infant inherits two broken copies of the primary power plant, they develop SMA. but they still have the SMN2 gene acting as a backup generator.
The tragedy of SMA lies in the specific flaw of that backup generator. The SMN2 gene differs from the primary SMN1 gene by just a single, seemingly insignificant nucleotide transition in exon 7.
Just one letter.
Just one letter. However, that single letter change alters the binding site for the cellular splicing machinery. When the SMN2 gene is transcribed, a repressor protein named HNRNPA1A2 binds to the RNA transcript and physically blocks the spliceosome from including exon 7 in the final messenger RNA.
Oh, wow. Because exon 7 is skipped and excluded from the blueprint, the resulting SMN protein is truncated, highly unstable, and rapidly degraded by the cell.
Exactly. The backup generator only produces about 10% of the functional full-length protein required. That 10% is enough to keep the fetus alive during development, but it falls disastrously short of sustaining the immense lower motor neurons after birth.
So the clinical severity of a child SMA is directly inversely proportional to how many copies of the faulty SMN2 backup generator they happen to have inherited. More copies mean a slightly higher accumulation of that 10% trickle of functional protein.
Right. And because the molecular mechanism of this splicing error is understood with such precision, scientists have developed therapies that directly intervene at the RNA and DNA levels.
The first breakthrough approach utilizes an antisense oligonucleotide, or ASO, such as the drug nusinersen. An ASO is a synthetic, short string of nucleic acids meticulously engineered to bind to a specific complementary sequence on the target RNA.
And the ASO therapy is designed to fix the backup generator. When injected directly into the cerebrospinal fluid, the ASO binds precisely to the site on the SMN2 RNA transcript where that repressor protein HNRNPA1A2 normally attaches.
So the synthetic sequence physically blocks the repressor from binding. By removing that blockade, the ASO forces the cell's spliceosome to recognize and include exon 7 in the final RNA transcript.
Yes. It tricks the faulty backup generator into manufacturing 100% full-length, perfectly functional SMN protein, halting the death of the motor neurons.
That is amazing. And the second revolutionary approach is AAV gene therapy, such as onisimnogene beparvovec. Rather than patching the backup generator, this therapy delivers an entirely new primary power plant.
Exactly. Scientists utilize an adeno-associated virus, or AAV, which is a harmless virus that is exceptionally efficient at infecting human cells. They hollow out the viral genome and replace it with a pristine, fully functional copy of the human SMN1 gene.
Billions of these engineered viral vectors are infused intravenously into the infant. The vectors cross the blood-brain barrier, infect the lower motor neurons, and deposit the healthy SMN1 gene into the nucleus.
And the new gene exists as an episome, completely separate from the host DNA, and immediately begins transcribing abundant amounts of the essential SMN protein.
Both the ASO and AAV therapies have radically altered the natural history of a disease that previously resulted in universal mortality in its most severe forms. It is a stunning triumph of applied molecular neuroscience.
It truly is. The chapter concludes its discussion of motor neuron diseases by briefly returning to infectious pathology, highlighting poliomyelitis.
Ah, yes. The poliovirus enters the body through the gastrointestinal tract, eventually invades the central nervous system, and specifically targets the lower motor neurons in the anterior horn of the spinal cord.
Right. It hijacks their cellular machinery to replicate, ultimately destroying the neurons and causing the acute flaccid paralysis characteristic of the disease, yet another stark demonstration of selective vulnerability to a systemic pathogen.
Okay, so we have thoroughly explored the devastating consequences of diseases that destroy the motor neuron cell body, the command center. But the biological machinery of movement can also fail if the command center is perfectly intact, but the communication wire connecting it to the muscle breaks down.
Yes, which transitions our analysis into the vast domain of peripheral neuropathies.
And the crucial clinical distinction here is that unlike ALS or SMA, which are pure motor neuron diseases, disorders of the peripheral nerves rarely manifest with motor symptoms alone.
Right, because peripheral nerves are bundled cables. A major nerve like the sciatic nerve contains thousands of motor axons transmitting commands outward, bundled tightly alongside thousands of sensory axons transmitting tactile and pain signals inward, as well as autonomic fibers controlling blood vessels and sweat glands.
Therefore, a systemic disease attacking the peripheral nerve will simultaneously impair both motor and sensory functions.
Exactly. The clinical presentation of a peripheral neuropathy heavily features sensory deficits. Patients frequently report paresthesia, spontaneous, abnormal sensations such as numbness, tingling, or prickling pins and needles feeling.
And as the sensory fibers degenerate, a patient loses the ability to perceive pain and temperature. This loss of protective sensation is profoundly dangerous, as it allows repetitive, unrecognized microtrauma to the feet, eventually leading to severe joint destruction and chronic ulcerations.
It is very dangerous. The spatial distribution of this sensory loss is highly characteristic. The text describes a glove and stocking pattern. The physical mechanics behind this pattern are fascinating. It occurs because the pathology represents a failure of axonal transport.
Right, a motor neuron sitting in the lumbar spinal cord has an axon that stretches all the way down the leg to a muscle in the foot. That axon can be over 1 meter long, making it the longest single cell in the human body.
And the metabolic logistics of maintaining an axon of that immense length are staggering. The axon contains no ribosomes. It cannot synthesize its own proteins. Every structural component, every enzyme, and every neurotransmitter vesicle required at the distant nerve terminal must be manufactured in the cell body in the spinal cord.
So these vital supplies are then physically hauled down the entire length of the axon along microtubule tracks by motor proteins like chysin, and the spent materials are transported back up to the cell body by dynein motors for recycling.
Right. When a systemic metabolic disease, a toxin, or an inherited genetic defect impairs this complex internal transport system, the longest axons suffer first. The distant terminals in the toes and feet are starved of essential nutrients and structural proteins.
The distal tips of the axons begin to physically degenerate and die back toward the cell body. Consequently, the sensory numbness first appears in the tips of the toes.
Yes. As the axonal dieback progresses proximally, the numbness slowly creeps up the foot and the calf. Eventually, the dying back reaches the slightly shorter axon supplying the hands, and the numbness appears in the fingers. The sensory loss physically resembles a patient slowly pulling on stockings and gloves.
And the motor deficits in peripheral neuropathies follow the exact same length-dependent logic. The weakness manifests distally first, resulting in foot drop or weakness in grip strength. The deep tendon reflexes, particularly the Achilles reflex at the ankle, are severely depressed or entirely absent due to the disruption of both the sensory and motor limbs of the reflex arc.
However, unlike a nerve that is acutely severed by trauma, chronic neuropathies rarely produce massive fasciculations, and severe muscle atrophy usually takes many months to develop.
Now, the clinical course of neuropathies is divided temporally into acute and chronic forms. The quintessential acute neuropathy is Guillain-Barré syndrome. This disease is an explosive autoimmune assault on the peripheral nervous system.
Yes, it typically erupts a few weeks after a seemingly mundane respiratory or gastrointestinal infection, often linked to the bacteria Campylobacter jejuni.
And the pathogenesis of Guillain-Barré syndrome is driven by a mechanism called molecular mimicry, right?
Exactly. The surface antigens of the invading pathogen happen to structurally resemble the glycolipids present on the myelin sheath or the axonal membrane of the patient's own peripheral nerves. The immune system generates antibodies to destroy the bacteria, but because of this structural resemblance, those antibodies cross-react and aggressively attack the host's own nervous system.
The clinical onset is terrifyingly rapid. The patient may notice mild tingling in their toes, which escalates over a matter of days into profound ascending motor paralysis.
The immune attack can strip the myelin so rapidly and extensively that the paralysis reaches the cranial nerves, causing facial weakness and ultimately paralyzes the diaphragm, necessitating emergency intubation and mechanical ventilation to sustain life.
Because the pathophysiology is fundamentally driven by circulating autoantibodies, the most effective acute treatments aim to physically clear or neutralize those antibodies from the patient's blood. We utilize plasmapheresis, a procedure that continuously siphons the patient's blood, mechanically filters out the plasma containing the destructive antibodies, and returns the red blood cells.
Alternatively, we administer massive intravenous infusions of gamma globulin harvested from thousands of healthy donors, which binds to and neutralizes the rogue antibodies, halting the autoimmune attack on the myelin.
While Guillain-Barré is an acute emergency, chronic neuropathies are vastly more common and have an enormous array of systemic causes. The most prevalent worldwide is diabetic neuropathy.
Right. Chronic hyperglycemia, elevated blood sugar levels, causes systemic vascular damage that chokes off the microscopic blood vessels supplying the peripheral nerves. Furthermore, the excess glucose enters the nerve cells and is diverted into the sorbitol metabolic pathway.
The accumulation of sorbitol induces massive intracellular osmotic stress and oxidative damage, systematically corroding the axonal transport machinery over years.
Chronic neuropathies can also be induced by severe nutritional deficiencies, particularly a lack of vitamin B12, which is critical for maintaining the myelin sheath. Chronic alcoholism produces a toxic neuropathy exacerbated by concurrent thiamine deficiencies.
Environmental exposure to heavy metals like lead directly disrupts the axonal cytoskeleton. Even systemic conditions like amyloidosis, where abnormal proteins deposit directly into the nerve tissue, or paraneoplastic syndromes associated with lung carcinomas, can trigger a widespread breakdown of the peripheral wiring.
To understand how these various insults actually cause symptoms, the text categorizes the clinical manifestations into negative and positive symptoms. The negative symptoms, the loss of sensation, the weakness, the absent reflexes are intuitive. The wire is broken or stripped of its insulation, so the electrical impulse fails to reach its destination.
And the failure of conduction can take two forms. In demyelinating neuropathies, the loss of the insulating myelin sheath drastically increases the capacitance and decreases the resistance of the axonal membrane. The electrical current leaks out.
Right, the action potential still propagates, but it travels slowly. This slowing destroys the precise temporal synchrony of electrical signals required for complex reflexes. If a burst of signals scattered across multiple fibers arrives at the spinal cord at different times, the reflex fails to trigger.
Alternatively, the signal can fail completely. The text references the historical work of the German neurologist Wilhelm Erb, who definitively proved the concept of conduction block in 1876.
Yes. Erb demonstrated that if he electrically stimulated a patient's damaged peripheral nerve distally, below the site of the myelin injury, the muscle contracted normally. The axon and the neuromuscular junction were functional.
But if he stimulated the nerve proximally, above the lesion, the muscle failed to respond. The severe focal loss of myelin physically prevented the action potential from propagating across the damaged segment. The signal from the brain was literally blocked in transit.
While negative symptoms represent a failure of conduction, the positive symptoms of neuropathy, the excruciating spontaneous bursts of neuropathic pain and tingling, represent a state of pathological hyperexcitability.
It seems deeply counterintuitive that a dying, damaged nerve would generate more electrical activity. However, the breakdown of the myelin sheath induces profound electrical instability.
Exactly. The denuded axon membrane reorganizes its sodium channels, establishing ectopic pacemakers. The injury site begins to spontaneously generate high-frequency bursts of action potentials in the absence of any physiological stimulus.
The loss of myelin also leads to a remarkable phenomenon known as aphaptic transmission. Myelin normally acts as perfect electrical insulation, ensuring that an action potential traveling down a large motor axon does not bleed over and inadvertently depolarize the small sensory pain fibers running tightly alongside it within the nerve bundle.
Right, when the myelin degrades, that essential insulation is lost. The naked axons lie in direct physical apposition. A massive electrical impulse surging down a motor fiber can literally short-circuit across the extracellular space and depolarize an adjacent, unmyelinated sensory fiber.
This aphaptic crosstalk generates a chaotic storm of false signals that the brain misinterprets as severe burning pain or intense tingling. The damaged nerve trunk becomes so mechanically unstable that simply tapping the skin over the nerve can trigger a massive, excruciating burst of these short-circuiting action potentials, a clinical phenomenon known as Tinel's sign.
Beyond acquired conditions like diabetes or immune attacks, there is a massive category of inherited genetic peripheral neuropathies. The text focuses extensively on Charcot-Marie-Tooth disease, or CMT, named after the three physicians who originally described it.
CMT encompasses a heterogeneous group of hereditary neuropathies that typically manifest in childhood or adolescence. The hallmark is slowly progressive distal weakness, severe atrophy of the calf muscles leading to an inverted champagne bottle appearance of the legs, and skeletal deformities like highly arched feet, known as pes cavus, driven by the chronic imbalance of muscle strength in the foot.
Researchers have classified the various genetic mutations causing CMT into two broad categories, relying heavily on the exact nerve conduction velocity studies we discussed earlier. CMT type 1 comprises the demyelinating forms. Patients with type 1 exhibit dramatically slowed nerve conduction velocities because the primary genetic defect targets the proteins responsible for forming and maintaining the myelin sheath.
When pathologists examine a nerve biopsy from a CMT type 1 patient, they observe a striking histological feature. The cross-section of the nerve is massively hypertrophied, and under the microscope, the individual axons are surrounded by concentric rings of tissue that look exactly like an onion bulb.
Yes, the onion bulb formation is the physical scar of a relentless, futile cycle of demyelination and remyelination. The genetic defect causes the myelin to prematurely degrade.
The Schwann cells, which are the cells responsible for wrapping the peripheral axons in myelin, detect the breakdown, proliferate, and attempt to repair the damage by wrapping a new layer of myelin.
However, because the structural proteins are genetically flawed, the new layer also degrades. Over decades, the Schwann cells lay down layer after concentric layer of failed myelin and connective tissue, producing massive, palpable hypertrophy of the peripheral nerves.
CMT type 1 demonstrates incredible locus heterogeneity mutations in entirely different genes on different chromosomes, all cause the myelin to fail, producing the same clinical syndrome. The most common variant, CMT type 1A, is caused by a duplication on chromosome 17 affecting the gene for peripheral myelin protein 22, or PMP22.
Right, and PMP22 is an essential structural component of compact myelin. An overabundance of this protein destabilizes the entire myelin structure. CMT type 1b involves a mutation on chromosome 1 affecting the P0 protein, another critical structural element that holds the myelin layers tightly together.
There is also an X-linked form of CMT that illuminates the complex metabolic support required by the myelin sheath. This form is caused by mutations in the gene encoding connexin 32. Connexins are proteins that assemble to form gap junctions, microscopic, physical tunnels connecting the cytoplasm of adjacent cells.
In the peripheral nerve, connexin 32 creates gap junctions that bridge the successive layers of the myelin sheath near the nodes of Ranvier. These tunnels allow essential nutrients and ions to diffuse rapidly from the outer surface of the Schwann cell directly across the compact myelin to the inner layers closest to the axon.
When a mutation destroys these gap junction tunnels, the inner layers of myelin are metabolically starved and slowly degrade. While CMT type 1 destroys the insulation, CMT type 2 destroys the wire.
CMT type 2 is the axonal form of the disease. Nerve conduction studies on these patients reveal perfectly normal conduction velocities because the myelin sheath is structurally sound. However, the overall amplitude of the electrical signal is drastically reduced because the internal axonal structure is physically corroding and dying back.
The mutations responsible for CMT type 2 target an entirely different set of proteins, those responsible for the internal logistics and structural scaffolding of the axon. For example, mutations in the KIF1 beta gene disrupt the kinesin motor proteins.
Right. If the molecular trucks responsible for hauling organelles down the microtubule tracts are defective, the distal nerve terminal is deprived of vital supplies and degenerates. Other mutations target the neurofilament light chains, the vital structural proteins that maintain the physical caliber and structural integrity of the axon. If the internal scaffolding collapses, the axon dies.
The text highlights even more obscure genetic targets causing axonal degeneration, such as mutations affecting ABC transporters located within the peroxisomes. Peroxisomes are organelles critical for breaking down very long-chain fatty acids.
Yes. If the transporter fails, toxic lipid metabolites accumulate and poison the axon. There are also mutations in proteins with incredible names like gigaxinin, which regulates the degradation of structural filaments. The failure of any one of these specialized internal systems ultimately leads to the same length-dependent glove and stocking degeneration of the peripheral wiring.
To conclude our analysis of peripheral nerve pathology, the text presents a fascinating condition that blurs the lines between genetic defects and acquired immunity, acquired channelopathies.
We typically think of ion channel defects as inherited genetic mutations. However, the immune system can spontaneously generate autoantibodies that specifically target and disable perfectly healthy voltage-gated ion channels on the peripheral nerve membrane.
A prime example involves autoantibodies targeting voltage-gated potassium channels on the distal motor nerve terminals. To understand the catastrophic effect of this, we have to revisit the basic biophysics of the action potential.
When a motor command arrives, voltage-gated sodium channels open, allowing positive sodium ions to rush into the axon, rapidly depolarizing the membrane and propagating the signal.
To terminate the signal and reset the nerve for the next command, the voltage-gated potassium channels must open. They allow positive potassium ions to rush out of the cell, returning the membrane to its negative resting potential. They are the essential physiological off switch.
In this acquired channelopathy, the rogue autoantibodies bind directly to those potassium channels and physically block their function. The potassium conductance is drastically reduced. The rapid influx of sodium depolarizes the membrane, but because the potassium off switch is broken, the nerve struggles to repolarize.
The axonal membrane is left in a state of sustained chronic depolarization. The distal motor nerve becomes wildly, pathologically hyperexcitable. Without the ability to properly reset, the nerve fires continuously.
This continuous barrage of electrical signals causes severe, unrelenting muscle cramps, massive visible fasciculations, and profound instability of the entire motor unit. It is a purely acquired electrical short circuit caused by a highly specific immunological blockade of a single molecular component.
It is stunning to consider the sheer complexity of the systems we rely on for the simplest movements. We have journeyed all the way from Sherrington's initial conceptual definition of the motor unit in 1925, through the precise clinical detective work of localizing a lesion with an EMG needle, down into the devastating molecular breakdown of the command center in diseases like ALS and SMA.
We have traced the electrical signal out along the biological wiring to see how failures in myelin construction, axonal transport, or ion channel repolarization manifest as profound peripheral neuropathies.
And it all reinforces the fundamental logic of clinical neurology. The ability to diagnose these conditions relies entirely on a deep, granular understanding of the underlying physiology. Because we understand exactly how normal conduction velocity works, how the spliceosome processes RNA, and how potassium channels reset a membrane potential, we can pinpoint precisely where the physical machinery is broken.
We don't just observe a weak limb. We measure an electrical potential or analyze a genetic sequence to visualize the specific microscopic domain that has failed. Unraveling these mechanisms not only allows for diagnosis, but is actively driving the development of therapies that intervene at the most fundamental molecular levels of the disease process.
Which leaves me with a rather provocative thought to end on. As we examine the breathtaking success of treatments like AAV gene therapy for spinal muscular atrophy, using an engineered virus to drop a completely new functional gene into an infant's central nervous system, it struck me that we are crossing a monumental threshold.
We certainly are.
We are physical beings. We interact with the universe solely through the contraction of muscle. All of human physical agency is mediated by this exact motor unit hardware. But with these targeted therapies, we are no longer merely managing symptoms. We are fundamentally hacking the biological hardware of human agency at the source code level. We are reprogramming the genetics of the motor neuron itself.
So the profound transition.
It really is. As our tools for neuroengineering become sharper, as viral vectors and antisense oligonucleotides become even more sophisticated and precise, how long until we transition from simply repairing a broken peripheral nervous system to actively upgrading the baseline performance of the human motor unit?
If we master the ability to manipulate the specific genes that dictate muscle endurance, the speed of axonal transport, or the recruitment threshold of motor units, the fundamental definition of human physical capability could be radically altered.
Something to mull over as you go about your day, relying on those millions of flawless, highly orchestrated action potentials just to stand up and walk across the room. That concludes part 1 of our deep dive into chapter 57 for Enkephalikos. In part 2, we will continue our journey down the motor unit, crossing the synaptic cleft to explore the neuromuscular junction, the autoimmune blockade of myasthenia gravis, and the complex diseases that destroy the muscle fibers themselves. We will see you then.